everybody. This lecture corresponds to the second part of excretory system. So let's have a look at my presentation, and here we go. Perfect. So in the first part of our lecture, we actually talk more about the osmotic regulation. And now we're going to talk about the excretory regulation of the different animals. And of course, how that is related with the osmotic regulation and how the different animal species and the different phylums cope with waste excretion. So as we know from the, we're going to pay particular attention to some actually components of the that are byproducts that are actually produced during the normal metabolism of fat, carbohydrates, and protein. So in the case of the fat and carbohydrate pathways, we normally will end up producing CO2, which actually is easily and normally eliminated through the lungs, through normal respiration, and water, which of course becomes metabolic water that is going to be used by the organism, or as we're going to see, it's going to be reabsorbed or excreted according to the osmotic gradient or osmotic situation, we can say, an equilibrium within the animal. But what happens in particular with dietary protein and the nucleic acids? Well, as we know, they are formed in the structure, they possess an amino group, which nitrogen normally binds very easily with hydrogen ions. So when NH3, which is ammonia, and NH4 urea start accumulating in the organism, in the body of the animal, that can actually lead to potential toxicity problems by an alteration of the pH gradient or pH situation or status in the organism, but also an increase in hydrogen ions can actually produce alteration in the electron transport chain. Remember that energy was produced in the electron transport chain within the mitochondria thanks to hydrogen or proton gradient that exists. If we have an excessive accumulation of ammonia and urea, then we can start having a problem. And ammonia will be actually more toxic than urea. So how do animals in general cope with this problem? Well, how do they get rid of this nitrogen that actually represent a potential danger or harm for the body? First, we see that most fish and other aquatic animals are mainly produce ammonia, which we see at the bottom is normally very highly soluble in water, and of course, and at the same time is also very toxic. But as the fish is of course swimming and is moving in an aquatic environment, by diffusion very easily, no much energy cost, at a very low energy cost, the animal will get rid of the ammonia, not really representing a problem. And of course, ammonia is going to be diluted by a huge amount of volume of water that represents this environment, so it won't constitute actually a problem. Mammals, like our situation, and other animals, we have the production of urea, which is kind of represent an intermediate state in terms of toxicity. And then, of course, we will need to get rid of that urea, and we'll see how. And finally, we have reptiles and birds, which are able to produce uric acid, which is not very soluble in water, meaning that it doesn't draw much water when it is actually excreted, which is good. That represents that these animals are able to save a lot of water, and that of course allows them to actually survive in really dry environment like desert, for example, in the case of some reptiles, right? And the case of birds. And also represent a big advantage of the formation or the synthesis of uric acid as a way of eliminating nitrogen, because uric acid it actually deposits as crystals in the amniotic egg, And that, of course, is a big advantage because those crystals, of course, represent no danger or harm or toxicity problems for the embryo that is developing within the egg. So that's also a really very smart way of getting rid of the excess of nitrogen by these organisms during the egg stage, during the embryonic stage, and of course not representing a problem. That's how these animals actually solve the excretion of nitrogen inside the egg. Now, what actually we need to retain and we need to eliminate from the circulatory fluid during normal metabolism and homeostasis of an individual? Well, we need to actually eliminate, as we saw, nitrogen, toxins, excessive water, in the case, of course, that we are having an excess of water, or high, for example, blood volume, an excessive or the excess of vitamins and micronutrients. So once we are actually covering the normal requirements of the animal, so the animal is going to get rid of the surplus of sodium, calcium, potassium, magnesium, phosphorus, and selenium. What the animal, or what we will retain from the circulatory fluid, well, form elements of blood like red blood cell, white blood cells like lymphocyte, neutrophil, eosinophils, and useful biomolecule breakdown products like amino acids and small carbohydrates. And also in blood, I forgot to mention, for example, immunoglobulins are proteins of a high molecular weight and size, and normally will be retained in the circulatory fluid. And of course, we need the immunoglobulins, so they are not actually excreted in urine. What we will retain under certain circumstances, like dehydration, for example, water, and vitamins and micronutrients when the body needs to actually absorb them, and the body actually needs to retain them. When we go and start looking at the comparison of excretory system structure between the different phylums of animals, we see that Porifera, Cnidaria, and Ctenophora do not have an excretory system present, so that's important not only for the lecture but also for the lab. But when we look at all the other animals in the throughout evolution, we see that all of them, from platyhelminths, Rotifera, Annelida, Mollusca, Nematoda, Tardigrada, Arthropoda, Echinodermata, and Chordata, all of them actually present more or less developed or more or less complex excretory systems. Now, let's start with a more simple one. Poriferas, as we said, they have an Cnidaria, they have Ctenophora, they have no excretory system. But in reality, this organism with a really low, we can say, metabolic rate, with of course, sedentary life, that they are not very active at all. And also they have a small body size, meaning that they do not actually require a centralized excretory system to eliminate their excreta. So that is also due to this really low metabolic rate they normally have, a minimal waste production. So they are not really challenged by being needing to excrete, for example, nitrogen compounds, like in invertebrates, for example, which can the retention, of course, or kidney failure will lead to a retention of ammonia and urea and ultimately end up causing the death of the animal. So Porifera and Cnidaria, they will easily waste excretion via first contractile vacuoles in the case of freshwater organism. But these contractile vacuoles, more than an excretion role or function, they actually fulfill an osmotic pressure status or equilibrium. So because they are, of course, living in a freshwater environment, meaning that the water surrounding the organism has less solute than the animal itself. So water will tend to get inside their body system, as we saw in the first slides of this, in the first part of this lecture. So of course, the contractile vacuoles will start growing as the water starts getting in the organism, and when it reaches a certain size, these vacuoles will actually eliminate this excess of water through the outside of the animal. And of course, when they do that, they also at the same time will excrete those wastes generated by the individual. The exact mechanism how these vacuoles actually work or draw water inside to be eliminated is not very well understood, but apparently they have proton pumps that actually pump protons inside the vacuole, and that will draw the water, and that's how they actually fill these vacuoles and how they get rid of the excess of water. And of course, they will excrete most of the wastes they produce by simple diffusion through the cell membranes, and also through the pores and the mouth or the integument. So it doesn't really represent a big challenge. And if we think about the marine forms, they are also isosmotic with the seawater. That means that they actually neither gain nor lose water in the body composition, so they are in equilibrium with their aquatic marine environment. Now, when we move to platyhelminths and flatworms, we find the appearance of an excretory system, which actually consists of what we call a protonephridia, which it is actually adopts a disposition of two highly branched duct systems that run along the body, and they're distributed throughout the whole body mass. And each branch actually ends on a flame cell, which actually is called a flame cell because they present a flagella that actually bathe very fast. And when they bathe, they actually resemble a flickering flame of a candle. That's why they were called flame cells. And that will create a negative pressure inside this tubule, and will draw water inside the cells, and then, the fluid will actually travel through a collecting tubule where water and useful metabolites are going to be reabsorbed and recovered back to the body. And nitrogen waste like ammonia will actually be eliminated, and it won't represent a problem because it will be eliminated through the integument of the animal across the entire surface. So this is more also a more kind of an osmotic regulation system than actually an excretion system. But anyway, for the first time, we have the presence of ducts. We have a fluid that travels through a duct, and in this journey, some substances are either absorbed while others are actually eliminated by the animal. So we see in the blue arrow how the body fluids are drawn in by the baiting of this flagella, resembling a flame. So water, nutrients, and other useful metabolites, of course, is going to be reabsorbed through the journey of this body fluid when it goes through the different ducts. In the case of Annelida, then we have the appearance of an nephridium, which is normally formed by an nephriospore, an nephrostome, which is an opening in one of the segments of the animal. It represents a ciliated funnel where the body fluids will get in, and then they will travel again through a net of tubule, a convoluted tubule, which is called nephric tubule. And it will actually wrapped, we can say, by a net of capillaries, and where by countercurrent, this water and metabolite absorption will take place in case of those useful substances that the animal needs to retain. And once the fluid is formed, it's going to end up in a bladder, which is kind of more widened tubule within the nephric tubule. And then we'll end up in opening to the outside in a nephridiopore, which normally opens in the next segment of the animal and will be wasted and eliminated to the exterior. In the case of arthropod, we have a different organization there, where they present an antennal gland, sometimes called the green gland for the case of a crayfish, and we will see this organ in the dissection of our crayfish. So the antennal glands actually are produced and ultrafiltrate forms by hydrostatic pressure at the end sac, And then we will see that the antenna gland will follow by a labyrinth, a tubule that will end up in a bladder, and throughout, along the length of this tubule, the fluid will experience a selective reabsorption and and will be active secretion, eliminating into the lumen of a tubule all these actual substances that the organisms doesn't actually need. But now, in the case of arthropod, they present what we call Malpighian tubules and rectal glands. It's a different excretory system than the ones we considered before, and the vertebrate kidney, in the sense that actually, rather than consisting of this absorption of the fluid and the secretion of components, this actually, in the case of this excretory system or arthropods, is different in the sense that the process is initiated not by an absorption process or function, but more by an active transport of hydrogen or protons inside the tubules. And by doing that, these protons will actually draw inside the tubules other ions by osmotic pressure, and that will actually start forming the, we can say, urine of the animal. So the urine formation is not formed by a process of filtration, but it's more formed by a process of tubular secretion by these cells that are actually lining the Malpighian tubules, and they are actually blind. So once the hydrogen is moved, or the proton is moved into the lumen, they will gonna be transported back to the hemolymph by transporters, but these protons, by exchange, will actually draw sodium, potassium, and chloride inside the tubule. Sorry, sodium and potassium, while chloride will actually follow passively. So that's how these actually do. So the osmotic pressure created by the ions will draw water, solutes, and uric acid wasted into the tubules. And in the rectum, water and salts are going to be reabsorbed by the rectal glands, and the end product will be uric acid, as we saw before, and the excess of water and wasted. So again, the concept in the case of the Malpighian tubules and the rectal glands is that the urine is not formed by filtration like the other excretory system, but rather by tubular secretion of ions are going to draw sodium and potassium inside, and chloride will follow passively, and also water will do the same. And of course, it represents an ideal system for dry environments, because the fact that it produces uric acid means that uric acid is not very soluble in water, meaning that it will not draw water into the rectum. So for this reason, this animal will save water, and that is, of course, an excellent adaptation to extremely dry, hot, and dry environments, like could be, I always put the example of a desert. Now, quickly going through the vertebrate kidney embryology, I want to make a couple of observations here. And one is that throughout the embryonic formation of the kidney, the reproductive tract is actually, reproductive system is actually, in mammals especially, is very close to the formation of the adult kidney. Embryonic and adult formation of the kidney. And actually, the reproductive system will actually end up sharing some of the ducts that the primordial kidney has or forms during the embryonic stage. So during the embryo development of amniote vertebrates, the kidney will go through a successive process or stages of pronephros, mesonephros, and metanephros. And the characteristic of this metanephros in, for example, vertebrates, will be that it's actually caudally located and is much larger and more compact than the mesonephros and pronephros. And they actually are characterized by presenting a very large number of nephric tubules. And now we have the appearance of a new structure in the metanephron, which is the ureter, which is going to be followed by the formation of a bladder and the urethra and to the exterior. So you can see there in the diagram of metanephros in adult reptiles, we have the testes and the epididymis actually using or having, or we can say, yes, using the ducts of the previous mesonephros to form the epididymis and the vas deferens as well. Now, let's have a look at the more complex and well-studied kidney of vertebrates. Urine in vertebrates is actually formed by three very well-defined physiological processes and different between each other, which is the filtration, reabsorption, and excretion. So the kidneys, which actually represent about 1% of the total body weight of the individual, receives 20 to 25% of a total cardiac output. So that's really a big volume of blood. So that allows the kidney to filter about 180 liters, that is between 45 to 50 gallons of fluid per day. But of course, most of these filtrate will end up being reabsorbed, because if not, the individual will very rapidly depleted of water and of course most of the important ions present in the body. So approximately in an adult human, urine will represent per day, the volume of urine formed in one day will be between 1 and 2 liters. That's between a quarter to half a gallon of urine per day. And that is using filtration, excretion, and secretion and reabsorption process done or performed by about 2 million nephrons. So we see that is quite a significant number of nephrons. So most, and about 99% of the filter is reabsorbed back to the blood. As we said, if not, will be very fast depleted of most of the water and useful ions and nutrients. And the other concept is that the kidney of vertebrates actually achieves a very high degree of control of retention and elimination of specific materials. The waste must be excreted in the urine, and the minimum production of urine calculated for an adult human is about 400 milliliters per day. That's the minimum amount or volume of urine we need to actually form if being able to eliminate our waste products. The functional physiological unit of the kidney is called a nephron, and we will see that during the travel through the renal tubule, both the composition by reabsorption and secretion and the concentration done mainly by water excretion of the original filtrate will actually change. So the original filtrate with the end product urine being excreted, they are completely different. They have a completely different composition and of course on volume. Now let's start with the nephron. We see that the nephron is actually first formed by the glomerulus, which is actually a tuft of capillaries, and the Bowman's capsule actually surrounding, wrapping this tuft of capillaries that are actually coming from the renal artery and drained by the renal vein from the aorta. These are branches of the aorta. And after we have from there, the presence of an afferent arteriole and an efferent arteriole, and we see what is important. So the nephron, so the renal corpuscle, is formed by the glomerulus and the Bowman capsule. And the nephron is the whole unit of the kidney composed by the renal corpuscle and the convoluted tubules and the net of capillaries and the collecting ducts. But we will see them one by one in detail. So we have the glomerulus, and the Bowman's capsule, both again forming what we know as a renal corpuscle. Then we have the proximal convoluted tube, followed by the descending limb of the Henle's loop. Then, of course, we have the ascending limb of a Henle's loop, which actually both the descending limb and the ascending limb of Henle's loop travel through the whole medulla, from the cortex to the medulla of the kidney. And we will see that that is fundamental for the formation of a gradient of solute, which will contribute to the modification and the absorption and secretion of ions, and in particular also the absorption of water that will contribute to the concentration and lead to the concentration of the urine in the individual. Then we have the distal convoluted tube, followed by a collecting duct. And in the renal corpuscle, as we said, we have an afferent arteriole, okay, coming from the renal artery. And this afferent arteriole, which is normally of a much higher caliber than or diameter than the efferent arteriole, that will actually produce extremely high hydrostatic pressure, and that hydrostatic pressure will be responsible for actually forming a fluid that will gonna be filtrated through these tufts of capillaries, and the fluid will be getting into the interior of Bowman's capsule, and from there will travel through the proximal convoluted tubule, and from there to the descending loop of limb of Henle's loop, the Henle's loop, and back to the ascending limb of Henle's loop, distal convoluted tube, collecting duct, ureter, bladder, and to the exterior. And we have the ureters there after the collecting ducts, followed by the urinary bladder, where the urine is actually, we can say, stocked, and then will be eliminated to the outside by urination. Now, we have here a detail of the efferent and the efferent arteriole. So again, there is a big difference, but it's not represented here, but it's a big difference between the diameter of the afferent and the efferent arteriole, which actually will produce the filtration of a fluid, mainly driven by hydrostatic pressure, but also there is a filtration barrier where all the form cells and form elements of blood will not pass through those pores that exist, and they will be retained in blood because, of course, they are necessary. So which are these components that will be retained? Well, of course, the red blood cells, white blood cells, then plasma proteins like albumin, like immunoglobulins, and what is going to go through this filtration barrier will be mainly electrolytes and small solutes, and that will produce a filtrate, which is a fluid we can define as almost devoid of proteins. So the process of the formation of this fluid inside the capsule is actually driven by hydrostatic pressure, as we said, by the difference between the afferent and efferent arteriole, and by a size-selective filtration through a filtration barrier. So remember, afferent is what gets in, and efferent what gets out. And this actually fluid will be formed mainly by water and wastes, but glucose, amino acid, ions, and vitamins, all of them or most of them will be actually reabsorbed back to the organism. So the conversion of this filtrate into urine will involve two processes. One is going to be the reabsorption of useful components of this fluid, and the secretion of wastes and modifying the original filtrate. And also will be a process of changing osmotic concentration of the urine by active water excretion, and we will see that how it works. Now, there is one concept here that is important to know, that is the concept of maximum transport, which is a kind of a threshold that the kidney has for the reabsorption of useful molecules. In this case, we're going to use as an example glucose. We see the glucose there that is in the filtrate of the glomerulus, but it's going to be transported back to the blood circulation because, of course, glucose is needed by the organism. And this is the situation where the concentration of glucose is less than 100 milligrams per 100 ml. Now, when glucose actually matches, glucose concentration in blood matches the maximum transport capability of the kidney or threshold, which is 100 milligrams per 100 milliliters. So we see that still the kidney can cope with the reabsorption of 100% of the glucose present in this fluid, in the fluid that has been filtrated into the fluid. But when the concentration of glucose in blood exceeds actually 100 milligrams per 100 milliliters, which could be the actual situation of diabetes, diabetes mellitus, for example, then the transport max of the kidney for glucose is going to be overwhelmed. I mean, it will be surpassed by the excessive presence of glucose. So some of its glucose will escape the reabsorption process by the kidney. And of course, we will have glucose present in urine, which is the what happens actually with patients or with animals that have diabetes mellitus. And the other thing I want to say here is that it's important to know is that each substance, each useful substance or compound for the organism has a transport max, a maximum transport threshold that of course varies with or changes with the substance we are considering. Now, let's have a look at the reabsorption of sodium. Well, the human kidney reabsorbs approximately 600 grams of sodium per day. That's a normal reabsorption of sodium by humans. So the amount of sodium that is being reabsorbed perfectly matches the amount of sodium that's being intake or is being ingested by the individual. So that's a way of keeping the homeostasis of sodium, the equilibrium of sodium within the organism. So let's put an example. If the individual is having the normal intake of sodium, which is should be 4 grams per day, then the net reabsorption will be 596 grams, and the other 4 grams will be excreted. So from the first number we saw, if we approximately the kidney reabsorb 600, so in this case it's going to be, because we are ingesting 4 grams, it's going to reabsorb only 596 grams. So the rest of the remaining 4 grams will be actually excreted in the urine. In the case that we actually have a much higher intake of sodium, just could be up, even sometimes up to 20 grams per day. And there are some cases where individuals actually get an intake of 100 grams per day. We see that the kidney actually is not able to excrete sodium as fast as it's being ingested. For that reason, sodium chloride will not be excreted, and that will produce a retention of water in the bloody fluids, and that's what causes one of the causes of increased weight gain in the individual. So it's actually surpassing the capacity of the kidney to reabsorb all the sodium that is getting into the system, and of course that will draw water in, and that will cause the weight gain. And that's the situation here in the US, where the ingestion of sodium per day is much higher than 4 grams per day. We saw that it was considered normal. Now, let's get into the kidney and see how the process happens. So let's concentrate now on the corpuscle and the proximal convoluted tubule. We see that water is going to be reabsorbed mainly by passive movement, while sodium is going to be reabsorbed actively, meaning by active transport, meaning through a cost of energy. So glucose, amino acids, vitamins, and nutrients like sodium, potassium, calcium, and phosphates will be reabsorbed using an active transport, meaning using energy, or facilitated diffusion. And in the case of water, water will actually follow the osmotic gradient, so it will follow the ion, so most of the water is going to be reabsorbed. So this transport is actually very tightly controlled and is regulated depending on the body needs. And some of this water is going to be reabsorbed by osmosis, again by passive transport as a different with sodium and chloride, and follow the solute reabsorption. Sodium, potassium, sorry, chloride follows passively the osmotic gradient. So 65 of the, at this stage in the proximal convoluted or proximal tubule or proximal convoluted tubule, 65% of the filtrate has been reabsorbed, and about 85% of sodium has been reabsorbed at this stage of urine formation. And now I want you to pay attention to something that is really important, that you can see that the fluid form in this part of the kidney, in the proximal tubule, closer to the cortex of the kidney, is isosmotic with plasma. So it has about 300 milli-osmolar concentration, so it's equal with plasma. But we see that as we start moving towards the inner medulla, from the cortex through the outer medulla into the inner medulla, we can see how the osmotic pressure, osmotic concentration, and solute concentration starts getting higher as we move towards the inner medulla of the organ. That will, as we will see, this gradient will actually produce, and this is key for understanding how urine is formed and how water is reabsorbed and some of the ions are excreted. We can see how in the distal or descending limb of the loop of Henle, how water is passively drawn out of this tubule because there are more solute that are present in the outer and inner medulla, which actually by osmotic gradient produces water to actually abandon this descending limb of the loop of Henle and start concentrating solutes inside the tubule, concentrated the urine, what is going to become the urine. So in the descending limb, it's permeable only to water. So that's a very efficient mechanism and very smart for start concentration the fluid. So the fluid increases its solute as we move downstream. But water, although it's the solute concentration in the fluid is becoming more higher and higher, how is it that water still is passively drawn out from the tubule? Well, again, this is caused by the presence of these gradients, osmotic gradients. So as we move towards the inner medulla, again, water, the gradient becomes higher, 4 times higher than the osmotic pressure of the plasma, which is about 300, and it goes up to 1200 milliosmoles, and that's how water still keeps being drawn from the fluid. So in the ascending limb, on the contrary, it's only permeable to solutes, and that actually causes by active transport in the ascending limb. Now we are talking about now solute like sodium and chloride is going to be actively drawn from the fluid into the medulla of the kidney, the outer and the inner medulla, and that actually contributes to the formation of this gradient that is by a countercurrent mechanism that is so important for water reabsorption and the formation of urine. So this high osmotic concentration in the medulla is actually created by an exchange of ions in the loop of Henle by current multiplication. That's how it is called. And this osmotic gradient is actually able to be conserved, so keep the medulla and the outer medulla and the inner medulla with a high osmotic pressure. This is done by the actual presence of vasa rectas, which are special capillaries that are located around the loops of Henle, and these vasa recta actually do keep the ions inside the medulla of the kidney. So the loop of Henle actually functions only in water reabsorption, but is of course fundamental for that. And in the loop, it is in the loop of Henle that 85% of the filtrate is actually reabsorbed. So again, we have two phenomena happening at the same time. On the descending limb of the loop of Henle, we see how water is actually being drawn or taken out from this fluid. And this fluid, of course, as it moves towards the medulla, is becoming more and more concentrated, always matching the osmotic gradient of the region of the kidney where it is actually traveling. And this water that is being drawn passively, thanks to this gradient of ions, will be taken away through the blood circulation. Now, as we reach the loop of Henle, we see a maximum concentration of 1200 milliosmoles. And then as we start getting into the ascending limb of the loop of Henle, we see how sodium and chloride is first passively drawn from the fluid, but as we start getting up or getting more towards the cortex, we can see how sodium chloride is actually actively being pumped from the fluid, and this sodium chloride and part of it will gonna stay close to the region contributing to this osmotic gradient that of course is used to concentrate the fluid by drawing water from the descending limbs of the loop of Henle. And then we will get into the distal tubule, which is permeable to sodium and potassium, will be controlled by aldosterone, which is a hormone which actually regulates that absorption, and by doing that actually regulates the osmotic pressure of the fluid and also of inside the organism. And in the collecting ducts have this particularity of being permeable to water, and the permeability of water is going to be controlled by the antidiuretic hormone of ADH, and this hormone actually when it's on high concentrations in blood, antidiuretic hormone, as the name implies, will lower the amount of water that is being excreted. That's why diuresis is the term we use for the excretion of water in urine. So if we have a high antidiuretic hormone, means that this hormone will actually produce the saving or the keeping of water. So it will produce a more concentrated urine. And this actually be the function of this hormone, either concentrating or diluting the hormone, whether in high or lower concentration of a hormone in blood circulation, will be done through the opening or shutting of water channels in the collecting ducts. And one thing that I forgot to mention that is really important here as well, we can see how urea is coming out of a collecting duct. So the collecting ducts are actually highly permeable to urea, and this urea will actually stay, it will be one of the solutes that's going to stay around the medulla of the kidney, and of course will contribute to the high osmotic gradient present in that region of the organ, and that of course will contribute to the passive absorption of water we saw in the descending limb of the loop of Henle, and of course in the loop of Henle. And to actually keep urea, the solutes around this region and contribute to this high osmotic gradient present in the kidney. This is also done thanks to the fact that the loop of Henle and the ascending first portion of a loop of Henle is actually non-permeable to urea, meaning that urea can be actually kept on that area and, of course, keep this gradient, which is fundamental for the formation of urine. So how does the loop of Henle actually change in mammals between species according to the environment where they live in? Well, the longer the loop of Henle is, as we saw in the previous diagram, so the longer the loop of Henle, so the longer will be, or the higher gradient of osmotic pressure we will have in the kidney. And of course, the higher the gradient, that means that more water can actually be absorbed. On the contrary, the shorter the loop of Henle, that means that the gradient is going to be not as high as other animals. And for this reason, not much water, they have less capacity of reabsorbing water, so they will be actually not able to produce such a concentrated urine as in comparison to those animals that have longer loop of Henle. So the longer loop of Henle allows more water to be drawn out and return to circulations. And in water environments, we will expect mammals to have longer loop of Henles or shorter loops of Henle. And we take the following example. The kangaroo rats in the Australian desert, they actually have really long loops of Henle. So that's the explanation why these animals are able to produce such a concentrated urine. And they are able to produce a urine that is 20 times more solid concentrated, more concentrated than blood. On the contrary, in animals that, for example, live in an aquatic environment like the beaver, North American beaver in the rivers, they actually present a really short loop of Henle. They only are able to produce a urine that is 2 times the concentration of blood. Now is the time to see the collecting duct. So we have the high concentration of urea that surrounds the collecting duct will draw more water out of the duct by osmosis. Then the water will get into the capillaries and will be taken away by the circulation. So again, we see that the amount of water reabsorbed and final concentration of urine will depend on the permeability of walls of the distal convoluted tubule and the collecting duct. That, remember, this permeability was actually controlled by ADH, or antidiuretic hormone, which is actually secreted by posterior pituitary. So there are brain receptors that are constantly checking the osmotic pressure within the organism, and that will actually give the signals for release or less production of ADH, which actually gonna regulate the number of water channels present in the distal tubule, and that will actually be produce a more or less concentrated urine. So ADH increases the permeability of the ducts by increasing the number of water channels in the collecting duct epithelial cells. So it's fundamental. And again, it is in the distal tubule that they are highly permeable to urea. And remember, urea, once in the medulla, cannot return to the ascending limb of the loop of Henle, so it stays in this region of the kidney, and that contributes to the osmotic gradient of the organ, which at the same time is the one responsible for most of the water reabsorption of the renal fluid. So 99% of the filter is reabsorbed in the distal tubule, as well as the waste products, and some of the water remains, which of course is going to constitute the volume of the urine form. And this is actually the homeostasis or the control of the water reabsorption by ADH. Let's start on the right. When we have a decrease in osmotic concentration in the blood, there will be a signal to the adrenal gland, which is going to be, sorry, going to be sending the signal to the pituitary, and the pituitary, posterior pituitary, will actually produce ADH, antidiuretic hormone, which is going to travel and decrease the permeability of the distal tubule and collecting duct. So less water is going to be retained. So that means then that there will be an increase in osmotic concentration of blood, and that will eventually lead to a dehydration situation. And then that will stimulate the receptors in the hypothalamus, and the pituitary, in this case, will release ADH, and that will increase the permeability of the distal tubules by this increasing the number of these water channels. So more water is going to be reabsorbed and it's going to be retained, and that will also decrease then in the osmotic concentration of blood, and so on. So you have here how the diagram is showing us how ADH actually is able to control water reabsorption in the kidney. So with this last slide, we finish with osmotic gradients, osmotic pressure, and excretory systems. So thank you very much for your attention.